Nicole is an assistant professor at the University of Alaska Fairbanks, and she's working at the Water and Environmental Resources Center, and I think you could read that in part of her bio. But she did a postdoc in archaeology, materials, and applied spectroscopy at the Idaho State. She specializes in marine ecosystems and is looking back history in ecosystem change. She also uses stable isotopes and elemental analysis. So I think her presentation today is going to be on the topic of using basically walrus bones. There's the cast of character, and using the bones as a way to look at the historic ecosystem of which those organisms were feeding upon. This is exciting to me since I work on the contemporary aspects of what they prey on at this time. I also know that people in the isotope lab here in Maryland have been looking at prehistoric horse bones for similar type of looking at ecosystem change and also prey base. So I'm very much looking forward to this, Nicole, and welcome.
Thank you. So yeah, this is a little bit of a change of subject, but it flows nicely with Paul's talk, because the whole reason we chose walrus, this changing sea ice and loss of sea ice, and how that does change everything from the bottom up. It's a bottom up forcing, and walruses are an important species to a lot of Arctic communities. And since I work in Alaska, it's important to a lot of those coastal communities. So we developed this walrus project, and it was adaptability and long-term responses of walruses. And it's a large group. And so here I just have the main players. And what I did with today's presentation, I just want to point out, because there's a lot of student data in here as well. So what I've done is put everybody's name down in the corner and their email. So, and this is online. So if you guys have questions beyond what you want to ask me, you're always welcome to email me, but you're also welcome to email the students and ask them questions, or email us all together. And of course, I just want to do this in the beginning because of the length of talks and stuff, but we want to thank all of our collaborating partners. We have the Eskimo Walrus Commission's been on board and fantastic. The St. Lawrence Island Subsistence Hunters have given, for 3 or 4 years in a row, they have taken samples for us, and that has included bone and teeth, which is nothing that anybody ever asked them before. So we went up there and talked to them, and they thought we were a rather odd group, which I've gotten used to because I'm always running around collecting bones, and people always think it's a little strange. And then we've had, obviously, the U.S. Fish and Wildlife, Alaska Department of Fish and Game. We've worked with the Rural Alaska Honors Initiative, which brings high school juniors and seniors down to the UAF campus. And in this case, some of them have worked in our lab with us and done small projects, which was great. So we were able to go back this winter and show off some of the students' work to the communities that they live in. So that was really great. And as Jackie said, what I like to do is actually connect our past to our present and our future, because I think there's a lot of information that we can get from odd places like museum collections and archaeological sites that can really give us insight into long-term change. And when I say long-term, for example, this walrus project stretches back about 3,500 years worth of data on walruses. And obviously we can't be as specific as if we go out there and, drop a net in and haul up a bunch of Arctic cod, every year. We just can't get that specific. There's all sorts of caveats, but it is still very useful information, and it helps people plan. We've had warm periods in the past, we've had cold periods in the past, and if we can figure out what was going on in those past periods, it may help us sort of plan for future, especially for, coastal or sort of co-management entities when it comes to these marine mammals that are so important, not just subsistence-wise, it's not just food, but for walruses, for many of these communities, the bone and the ivory carvings that they get are some of their only cash sort of economics that happen out there. So this loss of sea ice is a potential stress to walruses, which most of us know. On the Alaska side, we're getting massive land haulouts. This isn't the first time this has happened, but they are getting larger, and this can stress walruses out. This is also the land haulouts have happened a lot in Russia, ongoing, so it's nothing new for walruses, but it can cause some stress. And some of those things are, if you don't have ice to float around in and you're in one place, you may have to swim farther and farther for food as your season goes on. And that's an increased energy use. The loss of sea ice could cause changes to your food sources, which are a nutritional change. And there's increased disturbance just from shipping, from polar bears, from community interference. If someone goes out and passes a walrus haul-out, even just walking, and they all trample into the water. Some of the younger and smaller animals can get trampled to death, which we've seen happen. So there's all sorts of potential stresses in this loss of sea ice. And so again, we're looking at about 3,500 years of data. And we are getting genetics information from the bone, which will help us hopefully with population size and diversity over the past few thousand years. We are the first to extract steroid hormones from marine mammal bone. This was done a couple of times for human bone, but this is the first time anyone's attempted it with anything like this. And we're looking at both reproduction and stress hormones. We're getting trace element information from teeth, and that's helping us with foraging location and seasonality. We have the stable isotopes, which from bone and from teeth, which will help us with diet reconstructions. And we have been using some traditional knowledge, which sort of can help us with haul-outs, where they were, how often they were, traditional knowledge about sea ice conditions, diet of the walruses itself, and their ideas of population size and what's been going on with that. So today, what I'm going to mostly talk about is information that comes from bone. And walrus bone has what we call a very slow turnover rate. It's large bone, and so everything that we look at from bone is averaged over about a decade or more. And we're also looking at teeth, and the teeth allows us to look at closer down to seasonality. So all the cementum of the teeth, you can see these lines in here. I'll point them out. And these have to do with different seasons. The light seasons are growth seasons, and the dark seasons are, or the dark bands are, seasons with less growth. So you're basically looking at sort of simplifying it, a summer-winter seasonality. And then today, I'm also going to bring in some whiskers that we collected along with bone in our more recent stuff. And the whiskers bring it down to looking at very short amounts of time. So one whisker is probably looking at about 1.5 years of an animal's life. And so we can actually see close to like weeks or 2 weeks of individual feeding habits. So the first thing I'm going to talk about is cortisol concentrations through time, and this is in bone. So again, this is averaged over, minimum of a decade. So we sort of think of this as a lifetime average, and cortisol is a bioindicator of stress. It's sort of our fight or flight hormone. And we have both archaeological, historical, and modern samples from all of these. And what we can see here is that the modern and the archaeological walruses actually have similar cortisol concentrations, and it's our historical walruses. So from the 1880s to about the 2000s that are different than all of the others. And when we break it down for the historical, most of this difference comes from the 50s and the 60s, where there was significantly higher cortisol concentrations compared to modern day walruses. And let me minimize here we go. So the red line is actually an estimated population. So it's a mean, and it is estimated. Population of walruses is a really hard thing to nail down. But as you can see, populations did rise in the 50s and the 60s, as did stress. And what we're thinking is that the take-home message from here is that walruses actually may be more resilient to the climate change so far. In other words, the modern walruses aren't that stressed in comparison to our historic ones. And we think that stress probably stems more from very rapid population growth, or at least for what walruses is rapid population growth. So it's potentially stressful to have a lot of babies, and for the males, it may also be stressful to be, constantly battling with other males. Or so this is our thought right now, is that as of the data we have from 2013 to 2016, walruses are not stressed about this sea ice loss so far. We cannot see it as an average in their bones. This doesn't mean they haven't had stressful moments in their life, but remember, this is a lifetime average. And when you look at progesterone through time, which is their reproductive hormone, you can see as well that with a time lag, the hormones, the progesterone, the reproductive hormone, does shoot up as populations go up. But there's definitely a time lag for that, which is understandable given that, again, these bones, if you've collected it in 1960, they're averaging out what happened 10 to 15, 20 years in the past. So there's a lag, but they are definitely correlated with these population estimates. And they have lower progesterone and testosterone concentrations in the modern than the historic as well. And so this also tells us that there's a potential that we've hit carrying capacity right now for walruses, and that there's low calf production. So in other words, you're not going to get progesterone in a female if you're not actually reproducing, or you'll have much lower levels of it. So you've got a carrying capacity potential, and this sort of jives with what all of the traditional subsistence hunters have been telling us, that they seem to feel that there's less calf production recently, at least if you talk to people on St. Lawrence Island and stuff. So we're going to switch to stable isotopes. And the basics of this are that you are what you eat. And so I'm definitely over simplifying this, but on the left-hand side, you'll see there's lower trophic level to higher trophic level for nitrogen isotope ratios. And then on the bottom, you can see a difference between open ocean pelagic and shelf waters or benthic. And what you have here is sort of a increase in your trophic levels as you go up. So you've got an increase in nitrogen. And then, of course, our walruses are sitting over here because most of their feeding is done benthically. Most, but not all. So that's sort of an Arctic simplified stable isotope food web. And what we're doing is we're trying to combine this food web right here with information on sea ice data. And this is adapted from work from Anne de Vernal in the Chukchi, where she's reconstructed 6,000 years of increases and decreases in sea ice cover. And what we're trying to do is sort of take a look at what patterns of feeding looked like during past warming or past cold periods. So this is all very preliminary work. As I said, this is a lot of student work. We're still smack in the middle of our research, but this is 3,500 years of data. And what you can see is the green is the present, the blue are past cold periods, and the red are past warm periods. And what's interesting to us is, yes, the warm and the cold actually statistically are different, but the present, our data from 2013 to 2010 to 2016 is very, very different from everything else. And for those of you who know anything about stable isotopes, yes, this is all corrected. This is all SUS effect corrected. And in fact, we think we're actually fairly conservative in our SUS corrections. are big enough that we feel very comfortable with this data. And so, one of the questions is what drives this variability in walrus foraging? Is it location? Is it research change? Is it some sort of bottom-up driven factor, such as loss of sea ice and therefore a change in the balance of open water phytoplankton versus sea ice algae or something like that? So I did a preliminary small study with only 16 of our animals. Right now we have 550 or 600 individual walruses spread out over that 3,500 year time span that we've done stable isotopes for. But I took 16 and they were a good spread between archaeological, historic, and modern. And they also were quite spread considering what they appeared to be eating. So we have some that look quite high up on that isotopic food web. In fact, they look almost like seals or close to polar bears. And we have ones that look exactly as you would imagine they would look like if they were eating mostly clams. So I took a spread of those, just 16, and I looked at compound-specific amino acid analysis, which sort of gives us a baseline. It tells us what the plant baseline looks like for these. So these two up here actually appear to have terrestrial inputs. They're from two different areas, but they are both from the late 1800s. And what this may be telling us is that there may have been a lot of terrestrial input from some of the larger rivers at this day in these decades. And if you're pushing a lot of terrestrial matter into some of these bays, and the clams are filter feeding some of that, or filtering out some of that, that's going to follow up in the food web, and you would eventually see it. These guys here have mostly freshwater inputs. And that just means a lot of fresh water was coming into the system for these as well. And as you can see, one's archaeological, one is 2015, and one's 1995. So there's a lot more work to be done there. And of course, this is, as you would expect, all of the marine inputs. And what I need to do is I need to start some work on whether or not there's a separation in all of the marine inputs as well that we can break down to look at the difference between a majority ice algae, a majority pelagic phytoplankton type of, high sea ice level, low sea ice level input. One of the other things that is of interest is you can see the ones that are turning purple there on your screen. These guys, there may be some kind of geographic, even though walruses move all over the place, there may be some sort of geographic input as well, because these guys were hunted or collected in the same place, as well as these three here on the right-hand side. They're all St. Lawrence Island. It looks like two dots, but that's just because the 1930s male and the 2015 male from St. Lawrence Island are so similar that these points are overlaying each other. And the Kikulik archaeological sample also comes from right there on St. Lawrence Island. So these guys are all clustered together as well. So now we're moving on to tooth cementum and stable isotopes. And this is just starting. Alexander Thornton is a master's student who came on board 7 or 8 months ago, and he did some of this work for a poster he did for the recent Arctic Science Summit Week. And the basics to take away from here is that despite, again, a correction of our Seuss effect, there does seem to be an overall decrease from the 1940s to the 2010s, 2015 of carbon. And there may possibly be some in the nitrogen as well. And Alex has a long way to go with this, but one of the other things that he's beginning to see is a seasonal pattern potentially as well in some of these teeth. And you can see that in these increases and decreases in each individual tooth that you're looking at. And then I'm putting this whisker information up because this was a small side project that we actually did with those Rural Alaska Honors Initiative students. And it was a super interesting study, mostly because the general traditional knowledge is that seal eaters are few and far between. And for this project, we looked at 5 males and 5 females. This is the 5 males, and each one of these dots is us looking along the whisker at the stable isotopes. And what's interesting on this side where you see these circles is that all of these males were captured in the same place. And you can sort of see that last bit of feeding, that they were all eating the same types of foods. They cluster pretty closely there. But what's also very interesting is that one of the individuals was most definitely eating seals. You can see that they have a clear break in their nitrogen where it just comes shooting up, and then their carbon goes shooting down. And this is a classic sort of seal. Seals are eating fish, which are open water, so they have a reduced carbon signature, but they're higher up in the food web, so they have an increased nitrogen signature. And this is sort of a classic look of a seal. And so this individual was eating a seal, and you can see the exact opposite for them for clams. So clams are benthic, so they would have an enriched carbon in comparison to a seal, but they would also be much lower on the food web in comparison to a seal. So you have 2 very distinct signatures there for these animals. And this is a female. So this is also interesting because most of the subsistence hunters that you talk to will tell you that the seal eaters are males, not females. But here we clearly have, again, this signature where recently the female was eating clams, but at some point a year and a half, 2 years ago, she had definitely had an episode of eating a seal. And you can see this abrupt switch in the isotope values right there. And so we had one more animal in that group of 10 that had the potential to also be a seal eater. So we're looking at 2 out of 10 to 3 out of 10 animals collected in 2015 and 2016 that we've looked at so far with their whiskers who had seal in their diet. So this is a much higher percentage than we thought about. And the hunters have been really interested in this and also quite a bit surprised. So I'm going to move on again and look at trace elements and seasonality in tooth cementum. And there's basically a difference between the light and the dark cementum layers. And again, that light is probably late summer or fall, and the dark is probably late spring, late winter and spring. And you can see that the difference is not between the sexes, it's definitely between the seasons, and it seems to be driven mostly by zinc, lead, and iron. And so we're hoping that eventually this will help us sort of trace location by season for some of these animals. We don't expect this to continue to be so clear cut as we add more and more animals to this study, but this is the beginning of this portion of Casey Clark. He's our PhD student of his work on trace elements. So the other thing that we've done is look at traditional ecological knowledge with our partners in the Oral History Archives at the Rasmussen Library. And we managed to find 55 taped interviews that had quite a bit of information on walruses. And they were older tapes, they were in somewhat bad shape, and they've sort of been buried and not listened to for years. So we had a woman from Nome transcribe them for us, and she did a great job. We've digitized them, we've copied them for families, so not only did we bring them back to the communities, but we also copied them and gave them to the actual family members, some of whom knew this had happened but had never heard the tapes. some of whom didn't even know that their grandparents or great grandparents had recorded these. So that was just sort of a nice bonus and aside that we could do for the communities. And meanwhile, we are scanning through these transcriptions to sort of take a look at timing of the walrus hunt, sea ice conditions during the walrus hunt, et cetera, and the changes in all of these communities that we have the taped interviews from. And we're hoping to put that together with our decade by decade analysis for our historic and modern samples. The DNA, we have extracted and tested 83 archaeological samples, 80 historic samples, and 166 tissue samples from the 90s forward. And we are hoping to have all of that data well analyzed within the next few months. This is just something that's been taking a little bit longer as we wanted to carefully choose which samples we wanted to look at based on dates and data from isotopes and trace elements and hormones, et cetera. So this is trailing all the other research right now. So I don't really have too much to tell you about it, except that we definitely have really good surviving walrus DNA. And so this should help us out quite a bit in looking at those sort of population bottlenecks. We're assuming we'll see something during all of the height of the whale and walrus hunting in the late 1800s, and potentially something maybe during some of the warmer periods, but we'll have to see. So the project has had quite a bit in the way of broader impacts as well. We are really hoping that we're providing co-management and local community members with concrete starting points for discussions around their management. The local students participated in our rawhide research. We've visited the communities numerous times, 2 or 3 communities each winter, and we've done reports and presentations for them. We've returned those digitized and transcribed tapes to the communities and the family members. And last year, during the 2016 Arctic Science Summit, we held a workshop that was about the community, the community's research priorities. So we had fish and game and fish and wildlife there, but we also had flew in a bunch of community members, and we just sort of all sat around the table and said, what do you want to know? I mean, we're doing this huge project, but what else is there that you want to know? What's important for you to find out about with your subsistence, mostly based around walrus, but there was some talk about the seals and whales as well. And I have that workshop report. It's been sent to some of the funding agencies, but I could also upload it here if people are interested in seeing it. So our end result, hopefully, when this very large project is done, is to have a very broad picture of Pacific walrus adaptations to 3,500 years of change. I mean, we all know that everything's always been changing. So hopefully we'll have dietary change, both on the individual level and the population level. Hopefully we'll have an idea of foraging locations spread out over time and how those may have changed. We're hoping that we'll have all of this reproductive and stress response changes. Right now, we know that cortisol is highest during times when the populations are growing and not in our modern population, which is affected right now by sea loss. And so it may be that we are at a carrying capacity right now, and it may also be that as of yet, this loss of sea ice has not stressed the walrus's overall physiological system. If we do the same type of research a decade from now, that may look very different. But as of right now, it doesn't appear that the stress is long-term enough to show up in the bones. And again, the traditional ecological knowledge, we're hoping that will help us with changes in habitat use, prey selection, haul-out behavior, and we'll see what happens with our genetic diversity and population size. So I just want to acknowledge, again, all of our funders and all of our collaborators. And that's it. Thank you.
Thank you, Nicole. That was great. And I would just say that I would very much like to see that report, that workshop report that you did.
Sure.
Uploading it, probably, if you're willing to do that, that would be
Oh,
fantastic.
yeah. That was meant to go out to community, all sorts of communities, scientific. So yeah, that's great.
Sure. And it's fascinating about the seals. I mean, when we were out there doing IDA work in the Polinia south of St. Lawrence Island, I mean, that's when we documented the fact that the walruses were eating the seabirds, and so and at that point, the locals were saying that's why these rogue walruses have those yellow tusks. And so it's great that you're getting that feedback from the community too. But.
Yeah, and they were fascinated because they knew that they had added in at least one male that they identified as a seal eater. Mm-hmm. So now we're scrambling, going back through the paperwork, trying to find if that is indeed the male that we have.
Okay.
But they were shocked about the female. They thought they had only one in all of the 3 years that they've been giving us samples. And in fact, we have 2 or 3 at least just out of 10. And they've given us 30 samples from 30 individuals a year for the past 3 years.
So will you be able to then look back on time series on your bones and be able to say, it looks like it, that would there be more or less? And then, like, Bill, are they getting stressed enough that they're eating more seals than they would in the past? That type of thing?
It's possible. So I'm pulling this up now and I'm going to, can you see my mouse?
Yes. Yes.
2 dots up here.
Uh-huh.
This is 18 parts per million for nitrogen. So these guys actually look like polar bears, and that is in their bone.
Wow.
Which means these 2 here were consistently eating seals.
Right.
So, like I said, there's still more work to be done, but that was shocking to us. And if you look at this whole group here that I'm sort of circling, there is going to, in here, we think as we separate these out and look in more detail, there has to be some eider, maybe some fish. There has to be a mix of higher trophic level animals beyond the polychaete worms, which we know show up as a higher trophic animal and that gets sucked down by walruses on a regular basis.
Right.
So
that's great. Very interesting. Are there any questions from the folks on the line?